Great. Okay, we will start the webinar today. So good day, everybody. My name is Martin Beukema, and on behalf of the next generation of vaccinologists of the ISV, I welcome you to our monthly webinars. During these webinars, we aim to create a platform for young researchers to present their work, so the young researchers within the vaccinology field, but also to learn from experts in the field, from industry, for example, but also from academia. And today I'm very happy that we have two presenters from Australia. And so this is the first time that we will work in this specific time zone. And we hope with that we will also attract more young research from Australia and Asia. Before we start the webinar, I would like to share a few announcements. And therefore I will start sharing my screen. So today we will have two speakers from Australia, Sandra Depelsenaire and Alex Spence presenting. But before we start with these presentations, I would like to, really encourage you to become a member of the ISV. If you have not yet been a member, please join the ISV or renew your ISV account, because it actually comes with many benefits for young researchers within the ISV. So for example, the ISV organizes a mentorship program, but it also, let's say we have this group of next generation of vaccinologists, and only the mentorship of, let's say, a membership for 1 year costs only $35 for trainees such as postdocs and PhD students. So please, I encourage you to become a member of the ISV by joining through the link as you can see here in the top. Besides, I also encourage you to join the next generation of vaccinologists community. So what we do is actually we are organizing those webinars, and we try to actually connect all the different young vaccinologists within the ISV with each other. So we have a WhatsApp group, a LinkedIn group, and also you can find us on the website. If you screen this specific QR code, you can connect to our LinkedIn page. And if you would like to be part of our WhatsApp group, you can send us an email through the email address, as you can see here in the bottom. Please join us because we are actually a growing community. And if you would like to connect with other peers within the field, it's a great opportunity to meet others. I also would like to announce the next webinar that we have in December. So it will be on the 5th of December, where Dr. Ebony Gary from the Rister Institute will present together with Dr. Annie de Groot from APVax. With that, I would like to start off. I will stop sharing my screen, and I would like to start introducing Dr. Sandra Depelsenaire. Dr. Depelsenaire currently works as a manager at the preclinical department at Vaxxas, Australia. Originally, she's from Germany, but she obtained her PhD in biomedical engineering from the University of Queensland in Australia, after having completed diverse undergraduate degrees, and she spent a Bachelor of Science in Forensic Science, Toxicology and Molecular Biology, as well as a Honours in Nanotechnology, all obtained at the Murdoch University in West Australia. But prior to that, she also studied at the Ludwig Maximilian University in Munich, in Germany. So in her postdoctoral work experience at the University of Queensland and broad skill set acquired whilst working at the translational projects led to her current position. So Sandra's current work as preclinical manager focuses on managing a team of dedicated and exceptional aspiring scientists that are conducting R&D studies on new potential vaccine candidates, setting up new assays, prepare projects to go from R&D to phase 1 and beyond, as well as contributing to Vaxxas clinical trials by analyzing the clinical trial results. With that, I would like to give the floor to Sandra, and I hope you'll enjoy the presentation.
Thank you so much, Martin, for this kind introduction, and thank you all the attendees to joining and making the time for this. Today, what I would like to really share with you is how I did my journey from academia to industry, and how I worked on different continents, different cities, and different universities, and hopefully that might be inspiring or helpful to some of you. So hopefully you can all see my slides that I'm sharing. and I'll just get started. So first about the journey, a bit about the academia do's and don'ts based on my own experience, and also the industry, why not, and how important these next-gen vaccinologists, these seminars and communities are to really join them, and a little bit about Vaxxas also, where I'm currently working at. So first of all, about me. So I've done a PhD in biomedical engineering. So per se, you wouldn't really think that someone with biomedical engineering degree would end up in vaccine or vaccine research. However, I selected that using a very broad undergrad range from forensic science, toxicology, molecular biology, a diploma in biology in Germany, and as well honors in nanotechnology. These courses and degrees were acquired at 4 different universities. I've also worked at 3 different universities as a research assistant or as postdocs as well. And I've worked before that at the European Patent Office in Germany, in a bank, and currently also at Vaxxas. I'm also listed on various CIs as a chief investigator on different grants, and why I want to mention that is, irrespective of where you will be working, even if you decide to, for instance, consider industry, one can still be linked with the academia and pursue both avenues, which I'm currently doing, even though I work full-time in the industry. Throughout my early career research years, I've achieved over 25 manuscripts, book chapters, some patents, and attended well over 20 conferences. And other than work, I have 3 young kids that also keep me extremely busy, and some animals. And I do apologize, so English is my 3rd language, so if there are some mistakes, apologies for that. Well, important for me is also with the PhDs and the postdocs, so they can look differently. So for some of us, they can look absolutely beautiful. You see the bright light at the end of the horizon, and you know the clear path, and yes, it's hard work, you need to work your way up, but you can see the light at the end of the tunnel. In some other instances, it's a pure mess because nothing works. And whilst these two can be perceived as completely different, I think the key difference, or the main thing, is everyone learns on these journeys different aspects that will help all of us to become a better person, better scientist, better researcher, and better person in identifying better ways to do the science, which why we're all here. So for me, my journey has been a pure roller coaster. So when I started my PhD, it was a super steep learning curve. In the first year, when I finally reached it, it's something that I had to do a confirmation that was finally presenting in front of peers and colleagues and being questioned and grilled by examiners, which then led off like, oh, finally I've passed the first milestone, I can stay in my PhD. And the second and the third year were an absolute back and forth, a lot of changes in the supervisory team, change a bit of the project scope as well, which basically put me backwards several months. Then things worked well, then only to have another supervisor change that put me backwards again. Nevertheless, at the end of year 3, 4 for me, I could see the end of the light, and I've submitted, which was the epitome of those 4 years doing the PhD. But after I submitted the PhD, I realized I'm in the top 1-2% of the population. However, I'm literally at the bottom because I'm starting my postdoc and I don't know anything about what is after my PhD. And that was quite a daunting experience, and many of us, I believe, would experience something similar, because we get out of a PhD and then we have to look for grants, get on funding, and do the traveling, do all this networking in a more broader way that we have to do it ourselves. So for my PhD, what actually I worked on, and I think I want to highlight that because it will demonstrate and show you that no matter what you work on, you can really go from something completely separate into that is not related to your previous work, because you have learned the key skills, the technical skills, the soft skills, all of that make it together. So I was using a skin-based vaccine delivery device, a patch, to deliver vaccines into the skin, and then assess how these small projections that penetrate the skin could function as an endogenous or physical adjuvantation. So this work was at the time extremely novel. Whilst my background was very broad with nanotechnology and biology, toxicology, forensic science, I was suddenly working with vaccinologists, virologists, immunologists, but also because of that patch that we use, we had to work with engineers to understand how these work, how do we apply them, and material scientists to understand how's the interaction between the material and the skin, and how does that actually impact potentially the immune response downstream. I also worked with mathematicians who conducted modeling, so that I didn't have to do all the test parameters. So that enabled me really to identify what is my next question to look at and what gives me a higher probability or chance to address and understand what is happening. A lot of my work really involved the microscopy. So what you see on the right-hand side are images, for instance, that I acquired during my PhD, and some of them in the bottom right led to an award from the NHMRC. It's the National Health Research Council here in Australia. Irrespective of the analytical assays that I worked with, I learned in the end of optimizing assays, including the correct controls, and broadly being able to understand what is required from an analytical point, reproducibility, variability, and reduce the variation between operators or staff and team members. I also used a lot of the skin and cell-based assays, animals included, cell viability assays, and also flow cytometry in conjunction with the microscopy. So why is that important? Is once you master something, you learn on how to approach the challenges, the techniques, which most of us hopefully have successfully mastered during the doctoral studies. But not everyone is that fortunate, especially if some of these projects are really pear-shaped and nothing works, and whether it's gene expression, etc., these things can happen. So as I said, the key skills that I really gained for that were general lab skills, but most importantly also how to write and how to review. When I did my PhD at the University of Queensland, I was so fortunate to have a supervisor, the lab head, Professor Mark Kendall, who enabled us to really work very early on grants, to write sections of it, to review them, to reformat them, to generate figures, to generate the data, so that you know you have the space for one figure, and then you know there's the key figure that you need in it, so it needs to be clear, precise, without too much stuff around it, that you look at the figure and you understand what is actually happening. Also, through key skills that I acquired during my PhD was presentation skills. How to present complex matter in a very simple way, especially when you're talking to a mathematician who has no idea about biology, or to an engineer who doesn't really care about cells. You just need to convey what you need to get out of it. So that was an interesting experience, and I think that I've gained very valuable skills and knowledge in that area. But also I was working a lot on the administrative side, and what that means is, which I'll go through in the next few slides, is I was really focusing on what additional administrative stuff can I do. Join student associations, organize conferences, do inaugural student conferences, so that I could really gain exposure and experience on how to organize and run conferences, do the networking events like Martin is doing. And this is a fantastic opportunity. I'm so grateful to be able to present here today. Now, how did I get to where I got to and what was required academically? So first of all, I did a lot of student EMCR association work. I joined, and the acronyms that I'm mentioning here are specific to the Australian universities here. From my institute, I joined the student associations, the University of Queensland Australian Postgraduate Association. I became a conference co-organizer at the ICB&I, even where Bob Langer presented in 2012, 2014. I joined various committees in very small areas, and also, for instance, I'm also assisting with the next-gen vaccinologist. So it's really interesting and important. Join these committees because you get exposure to people, you get to know, get to meet them, and you never know who becomes a future colleague or employer for you. I also did many roles that acquired or that helped me to gain new skills: the secretary position, the treasurer, how to manage money for a whole team of students at the institute, how to manage the funds from a conference, how to write sponsorship brochures. So it was a fantastic opportunity really to gain these skills and still use them nowadays. I've also done a lot of volunteering work with regards to present not only in our team, but within the institute or within your school at the university on scientific topics, but also something completely different that is just really making me acquire these new skills. Myself, I jointly with other students, conducted the university open days. Depending on the country, that might not happen in every city or country, but we then took tours of students or potential future students through the university, the institutes, demonstrated and showcased our science. We also did some reaching out to the local high school so that we could really show to the students that could become the scientists of tomorrow what cool things we're doing in the lab, and how you can make the ice cream in the lab, and how you extract the DNA, not just from strawberries, but certain other things, and how you can see different chemical reactions. I was very fortunate in the lab, because it was so translational, to work with so many different people, and that led to many collaborations, working on Ebola, on dengue, on different mechanical engineering projects. So one of the highlights or the key feedback would be really where you can collaborate, reach out to people. The more you help others and work with others, the higher your chances also to get onto other publications. Do the science fairs, science days at schools, and even make a link between local schools, because that way students have a contact person, and that way, again, the institute and the schools can get more students. So again, all of these areas taught me key skills with the teaching, the supervision, and again, the reading, writing, speaking, sponsorships, how to deal with challenging situations, and writing proposals, basically. One of the key areas that I want to mention is I was so fortunate to work in a team with these exceptional scientists and engineers and mathematicians. We all worked extremely well as a team. So it never felt like you have to come to do work, you have to do 18-hour days. It felt like you go to work to see the friends, and you jointly work on something that you both, or 3 or 4 people, if you want to bring it forward. And it was not only what you know or you don't know, and also who you know, because if you know who might have the answer, that really accelerated finding the solutions to the problems. So what to prioritize? One of the key things that I think that is often underestimated is, if you're a student, you need to really look into what is required for my next step. If you're a postdoc, you might want to look into like, hey, I'm at level A, what do I need to do to get to level B? And seek out a senior staff member, a mentor, to identify where your weaknesses and what do you need to work on to get to the next level. Having that clear target in mind and in sight helps to have short and long-term goals, to reach them faster. Very often I find, even now while supervising students and doing mentoring work, many of them are not quite sure where they want to be, and it's really hard to help them if they don't have a clear path in front. Now, it's hard, of course, to have a clear path in front when there's so many opportunities with science policy and scientists and industry and teaching. I think sometimes take the time out to look back and just realize, have a look at a goal and investigate if there are options for placements, for internships that help to identify where people want to go to. Even in academia, it's so important to have publications. I think what has really helped me was aim for each year to have at least 1 publication or 1 review, and have at least 1 big impact paper from the PhD or the postdoc at least every 3 years, which is normally in Australia the length of a grant or a postdoc position, and use those to collaborate so that you can increase the numbers of publications that you can contribute to. Do extra teaching to assist with running some courses, lab courses, so that way you can emphasize and work on, you find students, potential students you can supervise, but it also gives you the skills to break down complex matter to explain it very simply. And here locally, what we have is also public speaking, where we do a 3-minute thesis competition. Present your whole PhD in 3 minutes, which is quite a challenge. And the same, the elevator pitch, 30 seconds. So really acquiring these skills will help all of us to become better scientists. So why the industry? For me, I was ready for a change. I know that some other people also want to have a job security and not that publish or perish. Some want to have a direct impact to the human health. And the other thing that also was important for me was a single goal, to reach really towards the same direction. I was fortunate enough to maintain my link to the university through a honorary position as an adjunct senior lecturer at the University of Queensland. Now, is, for instance, where I currently work, a PhD required? I would say yes and no. So it's really the position dependent. The key skill that we're interested in when we employ staff is, for instance, the lab skills, the management, and what do they bring on board? Because if you've learned an assay, you're normally capable of learning new assays quite fast. We may need to make sure that all the other aspects are readily available. Basically being a critical thinker and problem solver. There are many challenges in industry, which is like, it's very fast-paced, it can be translational, but more importantly, when there are challenges, we are very linked to the funding. So if there's an interesting question to answer, we sometimes can't pursue that because that is out of scope for our project. And we live and breathe acronyms, GMP, GDP, QMS. So all of those are on a daily basis. So that is maybe also a big difference to the academic world from what I found. Now, something very quickly, what I mentioned before, someone doesn't have to leave academia. Some interesting points could be internships or work placements. Why is that? In different work areas and workplaces, and even visiting a different lab could help you to have a different view, to see how they run the same assay in a different way with better outcomes, get to know more people. You could acquire new skills, and you also find out strengths and weaknesses in a new environment. Now, what I want to highlight is, as Martin had mentioned before, if you joined, for instance, the Next Gen Vaccinologist, the WhatsApp group, people would have seen that, for instance, Novartis advertised the next generation scientist internships. Now that is no longer accepting applications, but joining the team and this committee, or as a committee member, or just joining as a member, would be really helpful, that you can get these access to these jobs when they're posted. I also had a few years ago an honor student, which is basically an undergrad student, Raki, and she has done so extremely well that after completing her PhD, she was a finalist selected to do a R&D postdoctoral challenge in 2002, and she moved to Göteborg in Sweden to work now for AstraZeneca. So I think the limitations is really the horizon, and if you want to pursue something, reach out to people that you know, that you don't know, and ask how to help, how to get there. And most of us, if not all, are really willing and helpful to get there. So where I currently work at is Vaxxas. Vaxxas is a privately owned company here in Brisbane, Australia. And what we're working on is also a patch design to deliver vaccine into the skin. We have global partners, as you see on the top right-hand side. And the key difference from us, from different microneedle platform technologies, that we can also obtain a very robust dose-efficient responses because we can avoid the cold chain. We target also the pandemic responses, and we have conducted clinical trials with over 500 attendees, and we are really aiming to get to the late stage clinical trials and first commercial product in the future. We have various different departments, and for us the key thing is we all need to work together, and we are interconnected, whether it's in engineers working together with the analytical work people or the preclinical teams. We also manufacture, and we work with regulatory affairs, that's the FDA, to submit documents to the FDA and TGA. So what is my team and the department that I'm working in doing? So really we are looking, for instance, at targeting potential vaccine candidates and preclinical models, mostly rodents, but also larger animal models. We prepare these studies aiming for at least a phase 1 clinical trial, for which we will also need to run the GLP toxicology studies. So there's a lot of planning required, including a lot of documentation and paperwork. So that is the key difference for me, for instance. We work on the assays in-house, but not just like running an ELISA. We need to have reproducibility, reduce variability. The variance or the coefficient of variance need to be very low so that we can have batch to batch, very low variability. So this is when we run assays, is a much more stringent way than academic days. We work a lot with the other departments, as you've seen on the previous slide. And we work often in areas that might not be our area of expertise, but we acquire the skills because we have demonstrated we can achieve that. Now, we don't all need a PhD for these roles. It's really subject to what is the position, what is the requirement. But for instance, in our department, over 70% of the staff in our team have a PhD. Now, with that, I just want to stop there, say thank you so much for your attention. And that is part of our team that has now almost doubled since that photo was taken. So thank you very much for your time.
Thank you very much, Sandra. It was a wonderful presentation, and I think a lot of people could learn from your experiences during your career. For everybody who has a question, please put your questions in the chat box, and I will read the questions for you to Sandra. In the meantime, I have a question myself. So you were talking about mentors, and did you choose specific mentors because you wanted to achieve one specific goal, let's say one specific job, or were the mentors that just came across your path? How did you get to your mentors during in your career?
Okay, so one of the key things is the mentors take really like a lot of time out of their daily schedule to help the students. So for me, the key thing was really to make sure, what do I want to get out of it? So I selected the mentors for particular areas, and for instance, like, how do I get better? How do I improve? For example, public speaking. So then taking someone who has been aspiring or inspiring to me from networking events, just to acquire, talk to them, and just meet whenever it was feasible for them to acquire these skills. Now, for instance, I'm on the opposite side, right? So when I'm the mentor, I want the mentee to make the first step, to then say, hey, I need help with this, to assist these things. I'm also through the University of Queensland registered in the Women in Technology and Science fields, that I can assist them. But it's easier when the mentees reach out with a specific question, with specific areas that they want to override and overcome. And then from there, once it's started, if it's a good fit, that just takes on and goes forward. So that was most helpful for me.
I can imagine, yeah. It's indeed a very good point that the mentees reach out to the mentors. And with that, I also would like to advertise the mentorship program of the ISV that's offering. I think it's a great opportunity that has recently also been started. And I think for young researchers within the vaccinology field, yeah, there is an opportunity to get to mentors. Sometimes it's even difficult to get, I think, through your own institute, but there are opportunities. So yeah, I totally agree with you. If you have a specific goal in mind, try to find your mentorship with that. At the moment, I don't see any questions, but people, please feel free to put the questions still in the chat box. After Alex's presentation, we will also have the opportunity to come back to the questions for Sandra. So if you have questions in the meantime, please put them in the chat box. Sandra, thank you very much for your very nice presentation and for the opportunity to making the time, or for making time to present during our webinar. And then I would like to continue with our next speaker, which is Dr. Alex Spencer. Before we start with the presentation of Dr. Alex Spencer, I would like to introduce her. So Dr. Alex Spencer has obtained her PhD at the University of Sydney in Australia, before moving to the University of Oxford in 2006 to apply her knowledge on T cells on the developing of a liver stage malaria vaccine. During her time in Oxford, she was involved in developing new vaccine vectors, adjuvants, and optimizing antigen design and vaccination regimens, working across several vaccine programs, including malaria, influenza, Ebola, and SARS-CoV-2. So the translational focus of the Jenner Institute enabled Dr. Spencer to follow these approaches from preclinical studies through clinical trials, working with academic laboratories and industry partners. As part of the Oxford COVID team, Dr. Spencer performed a preclinical assessment of the Oxford AstraZeneca vaccine, measuring the immunogenicity across a range of animal species in combination with mRNA vaccines and rapidly testing new SARS-CoV-2 variant vaccines. So in 2022, Dr. Spencer relocated back to Australia to take up a lecturer position at the University of Newcastle and establish her own research group. So thank you very much, Alex, to present during our webinar, and hereby I give you the floor.
No problem, and thank you very much for having me today. I think it's actually worked out quite well, because Sandra's really touched on things that I haven't quite presented in terms of my PhD, but they're things I would have reiterated. So really, I guess what I'm going to talk to you a bit today is about my kind of experience, my very long 16-year postdoc that I did in Oxford, and then, which is a bit strange, most people don't stay in the same place for a very long period of time, and then kind of how I ended up back in Australia and becoming a group leader. So this is where I'm based now. Newcastle is 2 hours north of Sydney. Most people have heard of Sydney in Australia. Not many people have heard of Newcastle. We hug the coastline. I work at the university as a lecturer. So when I have to go on campus to teach students, this lovely Callaghan campus, that unfortunately has loads of mosquitoes. But my research group is actually based over here at the Hunter Medical Research Institute. And so that's the one research institute we have in Newcastle. In Newcastle, we have one university, one hospital, and we have one research institute. So we're really integrated here in Newcastle. Okay, so just a little bit of a background for me. So when I was in high school, I had a fascination for genetics and human biology. And when deciding what to do at university, I came across a degree in medical science. And I thought that would be fantastic because it was biology, but with a human focus. So I started my undergraduate degree. I took a year off between high school and university, and I did a Bachelor of Medical Science. And as I progressed through the course, my love for infectious diseases got stronger, and also I developed this real love for immunology. And actually already in my undergraduate degree, I was already fascinated by vaccines. So I went on to do an honours project. So in Australia, often the way the courses are designed is you have a 3-year undergraduate degree and then you have the option to do an honours project. And for me, that honours project was a 1-year research project. Again, really focused in immunology. I was working at a research institute that was associated, well, basically sat on the grounds of Sydney University. And at the end of my honours project, I loved doing research. I think I'd finally found my niche. And so I stayed on straight away and did a PhD again in immunology. So that took me about 4 years to complete. And when I finished my PhD, I'd already lined up a job at the University of Oxford. So I handed in my PhD on the Friday and then took up a job on the Monday at the University of Oxford. And I stayed there for 16 years. I went initially on a 6-month contract and came home 16 years later because it was really amazing opportunity and, you know, also lots of life events that meant I stayed in the UK. As mentioned, I came back to Australia in August 2022. I took up a position as a lecturer at the university and also establishing my own research group here at the Hunter Medical Research Institute. I'm fully employed by the University of Newcastle. It's just that through this 3-way collaboration between the university and the local area health, we have this Medical Research Institute where a lot of the university research is performed. So it's a slightly kooky situation. I have dual affiliation, but who pays my salary at the end of the day is the University of Newcastle. So just to give you an idea of my PhD journey and then how I ended up kind of in vaccines. As I said, I was based at this institute here called the Centenary Institute. That was really where a lot of the immunology research at the University of Sydney was being done. We had some big heavyweight immunologists, Tony Baston and Professor Barbara Vaxxas. And so I did my PhD in Barbara Vaxxas's lab, and I was really looking at T cells. So we had a lot of T-cell receptor transgenic mice that Barbara had made when she was a postdoc in Stanford. And we were really interested in these early events in T-cell activation, trying to understand what kind of drives initiation of responses. So I worked with a whole bunch of T-cell receptor transgenic mice. And really what I was looking at is how do these T cells compete with each other during an initial response? So if you take T cells from mice that recognize Henner-glycozyme and you mix them together with T cells that recognize myelin basic protein, do these interact with each other? Really what I did a lot of was I did a lot of mouse work. I did a lot of dissections, taking out lymph nodes. And I also did a huge amount of flow cytometry, and that was really where I developed my expertise in flow cytometry. The first day I arrived in the lab, okay, this is a long time ago. If you wanted to do 5 colors on the flow cytometer, you actually had to use the sorter. So the first day I was there, I quickly had to learn the dynamic, how the fluidics on a flow cytometer worked, which put me in good stead because now whenever there's a blockage on the machine, I usually can figure out where it is. My PhD was very much immunology focused. That was really the core of our institute. which meant that every meeting that, every annual meeting we'd go to would be the Australian New Zealand Society of Immunology. If I went to international meetings, it was the International Congress of Immunology, very much an immunology focused. So after 3.5, 4 years, I had a great experience. I love immunology, but I still always had this burning fascination with vaccines, and I always loved the impact that vaccines could make. So I moved from wanting to do something that was a very basic research into something that was more translational. And as I mentioned, I had lined up a job at the University of Oxford, initially on a 6-month contract, and the idea was I'd take my knowledge of T cells and try and apply that to vaccine development. So at the Jenner Institute, particularly, I was working with Adrian Hill, and he was really interested in making vaccines where cell-mediated immunity really plays a role, because in early 2000s, the licensed vaccines were really against diseases where antibodies were mediating protection. We didn't have any vaccines except for maybe BCG that really thought to initiate through cell-mediated immunity. And this is particularly the case for liver stage malaria. It's really clear that T cells play a very protective role. So we were working on strategies to induce T cells through vaccination and really kind of enhance these responses. At the time that I got to Oxford, they'd been working with a whole bunch of different viral-based vectors because it was clear that virus-based vaccines were much better in inducing T cell responses than, say, some of the traditional approaches like proteins. And just in 2006, they'd just started to work with adenoviruses. And importantly, I was working initially with a focus on liver stage malaria. It kind of moved into testing lots of different platforms. But the way that the Jenner Institute operated, it meant that whatever we were doing on liver stage malaria got applied to lots of different vaccine programs because we were extremely integrated in the Jenner Institute. And it's because if you think about the way that we look at vaccine production, we'll have a lot of work done on vaccine design. And then the typical thing is you do preclinical and immunology efficacy. You go on to make your vaccine, and then you go through clinical trials. And a lot of, what happens in phase 1 feeds back into your vaccine design. And if you were just in a single lab looking at, say, development of a malaria vaccine, really probably what you're focusing on is just the strategies that will get you across the way. But as I say, at the Jenner Institute, we had lots of different multiple programs running. We had, when I first joined, it was tuberculosis and malaria. It was a lot primarily on liver stage malaria, but also then blood stage malaria. The malaria program expanded to also eventually including transmission blocking vaccines. We also started looking at it in influenza. So essentially we ended up being a vaccine focused and with different groups working on different specific diseases, but actually we were all integrated. So anything that we were doing in vaccine design kind of crossed paths. And as I say, it was a really fascinating place to work, and you definitely got an understanding of the entire vaccine development pipeline. So initially we were in another building, then we actually brought together everyone that was part of the Jenner Institute into the Jenner laboratories. So we were based in this little green building here. This is a map of the kind of precinct. So it's one of these funny places. We have one of the hospitals in Oxford, then you also have the University of Oxford, which is right next door. And we were on the Old Road campus research building here. We had the Wellcome Trust Centre for Human Genetics. To be honest, there's a lot of new buildings over here, but we moved into this particular green building here. And here we were doing vaccine development. We were doing preclinical studies. But importantly, we were also sharing space with people doing the immunology on the clinical trial. So at this point, we were all starting to work together. It meant that in the labs, even if you were working on preclinical studies, there were parts of the labs that were working at GLP and GMP grades. So that's good laboratory practice and good manufacturing practice. HTA is the Human Tissue Act. So there was a lot of regulations and a lot of information that people needed to be aware of. What we were very lucky in Oxford as well is we had the Centre for Clinical Vaccinology and Tropical Medicine. And this was actually where the clinical trials were performed. So we had some staff members that were based over here that were running the clinical trials. So these were the clinicians and also the project managers. And it was a walking distance. So it was a 15-minute walk, 5 minutes if you drove. If you cut across the car park here, you could make it in a bit of time. And actually, we would have lab meetings. We'd alternate. We'd have lab meetings over at the clinical trial area, or we'd have labs at the laboratories, really trying to make sure that everyone in the Jenner Institute had good global knowledge of what was going on lots of different programs. The other amazing thing that we had in Oxford is there had been this clinical biomanufacturing facility that was set up, to make antibodies. And we kind of took control of it, took it under our wings. I can't remember what year it was about, probably about 2006. And because this was a GMP facility, it meant that we had easy access, and we actually had control to make small grades of a clinical stock of our vaccines. So we started manufacturing adenoviruses, and then when we started working on the pre-erythrocytic vaccine R21, we also did the manufacturing there. It also meant, so again, you got to understand what was required in terms of manufacturing, but you were also readily available to do some of the potency assays. And that meant that my time in Oxford actually got to be involved in lots of things across the pathway. So I did a lot of work here in vaccine design. Again, things would switch over. We'd often do collaborations with companies, depending on the kind of the projects we were working on. So to start with, in the first project that I was funded by, we worked with Cambridge Biostability and Acaris and Amaxio, looking at the ways that we could incorporate their technology into our vaccine design. We also did a lot of preclinical testing, and obviously depending on the disease that we were working in or the kind of the particular focus of the assay, we worked with lots of different animal species. So we did a lot of work with mice, primarily in Oxford. When we were looking at taking an adjuvant forward into that would go into human clinical trial, we originally needed to do some safety studies in non-human primates. So we didn't house the non-human primates in Oxford, but we sent vaccines across to the US and got the samples back. So I worked with processing non-human primate samples. We also, because we had an influenza program, we had collaborators at the Pirbright Institute that works with chickens and also works with pigs. Because the Jenner Institute was a collaboration also with the Pirbright Institute, which was all about One Health, it also meant that sometimes we got involved with studies in cows and some in goats. So kind of got to tap into lots of different areas of preclinical testing. As I said before, because we had that clinical biomanufacturing facility, often involved in potency testing. So I had to have a really good understanding of GMP, but also kind of rules and regulations around SOPs and writing release assays. We would ship our pox virus vaccines to IDT in Germany, and then they will come back, but also we'd have to have lots of testing of those. We had vaccines that went to a manufacturing facility in Italy and then would come back for testing here. When we started with our malaria vaccine, we started working with the Serum Institute of India and Novavax, and again, starting to work with these industry partners. And I got to see a little bit about how different companies worked, and particularly got a better understanding of what would be a focus in an industry partner. We ran clinical trials. We ran clinical trials in the UK, but also we ran them overseas. And sometimes I was involved in some of the immunology of the clinical trials, both in the UK and then in our partner sites. And so this is just thinking about my long PhD journey. I put this in just to kind of demonstrate that I really kind of, in my time in Oxford, I moved from junior roles to senior postdoc roles to more senior roles, and kind of with that taking on more responsibilities in that time. So initially I came to do a lot of preclinical immunogenicity studies, and then I started to supervise and train people on those particular assays. I was had a lot of experience with animal handling, particularly in mice, and so when there were new students that would come along, I would often teach them techniques. Because I had expertise in flow cytometry, I would also help out on the clinical trials. And, you know, it's one of these things, once you start working on something, you kind of never really stop. So in about 2009, I started to manage really all the mouse work within the Jenner Institute. So that got me some management experience, but it also got me to really understand the workings of the regulations in the UK. In about 2015, I also took over a management of our insectary, which was all about rearing and maintaining a colony of mosquitoes and providing mosquitoes, malaria-infected mosquitoes for work. With that experience with mouse work, I was asked to sit on the Clinical Medicine Ethics Committee. At this time, that was a great opportunity because I got to see the different workings within the university. When I took over management of the insectary, I sat on the Institute Safety Committee, but also because we then started to run it as a containment level 3 facility, I also got a bit of understanding of the workings of that. I had lots of different fundings. None of these were typically straight for me. So when I first came, I worked on a project that was funded by the Gates Foundation. When that finished, I switched over to some funded from Wellcome Trust. At some point, I moved over onto a project that was European funded. But sometimes, so 2017 onwards, I had a lot of one-year contracts, lots of short-term contracts. So I would say that I was at Oxford for 16 years, but I never really had a contract for more than 2 to 3 years, which is unsettling, but at the same time, I kind of was in a place and doing lots of activities that everyone wanted. So they you know, always found some salary for me at the end. As with growing on my senior responsibilities, I also took on supervision of students. I worked with some really amazing graduate students over the years. Some worked on independent projects that I started to do. Others were kind of in this global projects that were being performed. And the other thing I was going to say is I also had a few life events that came along. So in 2012, I had my first child. This is me probably about 2 weeks before I went on mat leave with a lab coat on because I was trying to get everything I possibly could get done before I went on mat leave. And my second child was born in 2015. So taking on a lot of management jobs and lots of different projects and also juggling life at home. It was about when my son, when my daughter, so I have a daughter and a son. When my daughter was about 2 years old, I decided I really wanted to get back into immunology. I had really been focused on kind of the Jenner Institute, and I missed immunology. And so they put a call out in early 2015 for people to join the Oxford Immunology Group, which I did. And that was a really great experience because I got to see and network with other immunologists in Oxford. We organised local meetings. We also eventually, the university developed an immunology networking group. And because of that experience organising local meetings, it meant I felt I then took up a position with the British Society of Immunology in 2018 to be part of their Congress committee. So this is, I would say, at this point here, I started to actively make time for my career and really look at other opportunities outside of just getting some work done. There were some really great opportunities I had while I was at Oxford to work at collaborator labs. Sometimes they were specifically related to my project and sometimes they weren't. So as I mentioned before, we had often worked with biotech companies. In 2008, we wanted to demonstrate that the adjuvant we were working with could enhance responses, and perhaps we could do this through electroporation. We couldn't do it in Oxford, so we decided to go to our collaborator site in Italy to perform the experiment. It was a really great experience because not only did I get to experience the Italian summer, but I got to see how a different laboratory operated. As I mentioned before, we had some rhesus macaque studies that were occurring in the US, and at one point we needed to run flow cytometry on some fresh samples, and so I went to the US for 2 weeks to work in a containment level 3 facility, which is how they processed all their samples. It was really fascinating. I happened to be there at the time. It was 2010, Obama was had his motorcade came through. Anyway, I got to see some very interesting US politics. That's probably not the same today. The key point being that even when it was project related, I took up opportunities to go and work in other labs. I guess my expertise in flow cytometry enabled me to also go to work with partner sites, often that wasn't quite project related. So because of that integration between preclinical and clinical, when we started to take our malaria vaccine to malaria endemic sites, we really wanted to set up flow cytometry assays on site. And so I got asked if I could go to Kenya and if I could go to the Gambia to really teach staff there how to use our particular protocol to run fresh flow cytometry. So I spent a week at Kemri, which is on the Kenyan coast. And then another time I actually ended up going to the Gambia twice. The first time was to teach them technique. And then because one of our critical time points was occurring in Ramadan, they really needed some extra hands to help. And so I really put my hand up to say I wanted to go there and have a look. And it was actually it was a really great experience. I got to see what the field sites were like, but also working with some really highly motivated young researchers. And I think they had a greater motivation to do science than some of the students that I supervised in Oxford, I would have to say. But I guess as I mentioned before, I had a lot of short-term projects, and I guess that was because I became a little bit indispensable into the institute. So I took on kind of roles and responsibilities that were essential to operating of the Jenner Institute. So I managed all the mouse work within the Jenner Institute. It started small and then it got bigger and then it got bigger and then it got bigger. So I kind of took the responsibility for training staff and submitting annual reports and applying for new project licenses. And I was kind of this main point of contact between Anima facility and our researchers. As I said before, I took on managing the insectary. It was primarily because I needed mosquitoes to start with. There was no one else that could take it on. But it was a really good experience because we ran it as a little small research facility. So I got experience costing and planning and invoicing, and I kind of ran it as a little small company. What I also had a lot of experience with was doing vaccine potency assays. So here is when our manufacturing facility made a clinical grade vaccine and needed to demonstrate that it was immunogenic. It got me to understand the process of releasing vaccines and thinking about GLP and report writing, but also I got experience working with industry partners and then I would get a better understanding of what would be the focus of their what they were trying to get out of these particular studies. I would have to say it did keep me in a job, but at the same time, it probably held me back a little bit because if you're a really busy person, you focus on getting the work done. And when you get things done, people often don't want you to leave that particular job. So yes, it kept me in a job, but at the same time, I didn't really get pushed to develop my own independence. And that's probably why I stayed as being a postdoc for 16 years in the same place. But in saying that, that experience working across lots of different animal models, doing things quickly, working clinical, preclinical, robust assays, it meant that when COVID came along, I had the right expertise and skills that I could quickly switch to supporting the COVID vaccine development. So it happened to have been in Australia in the start of 2020. Actually, January 2020, just before I went on holidays, I ordered mice for them to perform some studies before I got, while I was away. I got back and quickly got pulled into a meeting. I think 2 days after I was back from holidays, I was working full-time on COVID vaccines. Suddenly I was processing ferret samples, never processed ferret samples before. I was processing PBMCs from rhesus macaques, really because I'd had that experience working across lots of different bits and pieces. And so, as I said before, I came back to Australia. My husband is Australian as well, and we've been really looking for opportunities to return to Australia. He had his own business and I had a pretty secure job. So we really wanted to have some firm reason to be coming back to Australia. One of us had to have a job. We had been looking, but COVID kind of disrupted our plans or kind of made us think otherwise. I had, at the time in Oxford, been starting to develop some small research projects. I had been applying for small pots of money. In my spare time, I was trying to write investigator awards, which were kind of not really being all that successful. I'd also been doing a bit more teaching in Oxford. I'd been doing some Masters of Immunology program. And then, you know, when I stopped to think in the middle of COVID, got really decided I need to get back and focus on where my career was going as opposed to just kind of getting jobs done. I got a mentor through the British Society of Immunology in 2021, and he kind of opened up the idea about taking on an academic position and teaching. And so at the same time, I did this course, which was learning and teaching, and became an advanced fellow. And as I said, I got a job in 2021 and I moved back to Australia, to Newcastle. And so the way that the academics it's called an academic workload here at Newcastle, is you get a 40-40-20. So I spend 40% of my time doing research, 40% teaching, and 20% doing service. That research is really kind of establishing a group, doing ethics, safety grants, and supervising research students. The teaching is doing course coordination for undergraduates, running lectures and workshops. And that service is kind of all of those things that we do in science. So manuscript reviews, sitting on committees, being a conference organizer, examining PhDs from other universities. I mean, we say it's 40-40-20, but I would say that's probably like 70-70 and 30. So it often feels like a two-person job. My focus these days is really looking at how I can use my experience working with vaccines plus my love of immunology and that really basic question of how does the immune system work. I'm really looking at how do different vaccines drive these different responses. We know a lot in vaccines about what happens here. We're not so certain about what happens in between. So what I'm really trying to set up is looking at how vaccines, how different vaccines can alter antigen presentation and then alter the immune response. I can't remember what I put here. Since returning to Australia and setting up my own group, it has been a very, very steep learning curve, and I feel like I've not really made a lot of progress. The teaching has taken a lot more time than I hadn't quite realized it would. There's been a lot of, you know, we have course coordinations. A lot of things are really on a time critical nature. You need to respond to students quickly. If, you know, my job at the university is dependent on how good a teaching score I get. So, you know, you do want to be responding to students. You also want to be looking at ways to really optimize the learning of students. And I'm also setting up the research. I've had to do all the paperwork, so ethics applications, safeties. I spent a long time running grant applications. In Australia, we tend to have the main funding bodies just take one grant application a year. So there's a long time between grants. I think I'm up against it because I've spent a long time abroad. I've had to relearn how to write Australians in grants in the Australian context. Also re-established myself as a researcher in Australia. I think I had started to build my reputation in the UK, but then coming back to Australia, I've had to rebuild that. So establishing collaborations and also recruiting students. It's really only been through teaching in the undergraduate courses that I've started to recruit students, and now it's looking at the funding. So I guess my long 16 years has not been the full. I think you see people that have a much stronger trajectory, but my little tips would be that always say yes to opportunities, be open to new experiences because you never know where they're going to lead. Learn new techniques where possible. I think it's really important to try and experience a different laboratory. And I know that we traditionally would have said that you start your postdoc and then you move overseas, and that can't, you know, personal circumstances mean that can't be possible. But perhaps a way to really do that is to do a short-term placement or just looking for ways to just do, you know, a week or 2 weeks in a different institute or a different lab. I think the key is make connections with good people who are nice and they respect you as an individual. They do very good science and they want to pass on their skills and knowledge. And this is my big thing. In a world where you can be anything, be kind, because kindness and being nice to other people is really having connections with people is how you feel happy. And this is just to say there's definitely the connections that you make along the way. that's really enriched my life as a scientist and helped me to have some really great networks. I had to just be cautious of what photos I put up on here, but you know, there's people that I've worked with have gone on to do some quite amazing things. And it was really a really wonderful environment where I was a postdoc. And thank you for listening.
Thank you very much for your very wonderful talk, Alex. We already have a few questions in the chat box, so I'm just going through that because since we're already we're already at 10. So there are actually already 2 questions about actually how you establish your work-life balance. So it's being a mother. How do you combine that, and did you see any challenges and hurdles when you combine motherhood with an academic career? How did you overcome them? And do you have any advice for the young researchers?
No, I wish I did. Sandra might also like to comment here. I had a student this week ask me how I balance it all. This week I'm not balancing it all that well. There's a lot of burning deadlines at the moment. Having a very understanding partner. I would definitely have to say having kids that are very resilient and also very understanding. So in COVID, when both my husband and our work got very, very crazy, my kids were great and they knew what the focus of the project was. And so they would just be very happy for us to go to work. So I managed to develop some resilient kids at the end of it. I think it's always a challenge. I am yet to find the balance. I hope Sandra has perhaps got a word of comment into you, perhaps.
Yeah, it's just gonna.
Words of wisdom is just like, try your best, be kind, and try to teach the kids resilience. So that goes a long way.
I think that's great. Just another question, maybe for both of you, and it's again about a mentorship. So how can a student go about finding a mentor? So you already mentioned, for example, the community that you were a member of, but do you have any additional tips for?
So I am a person that doesn't like to ask for help. So I have never really found a mentor. So I actually went through mentoring programs. I felt for me that it felt more structured. It also means that the person who signs up to being a mentor has actually actively engaged in the process and therefore is going to be really prepared. So if you're too afraid to go and ask someone, try and find a mentoring program to start with, because that will build your confidence to then go and find a mentor external to a program.
Wonderful. And
I think Alex made a great. So I think
ahead.
Alex made a great point there. Just if I can add to that with the mentor, I think most important is the students or anyone needs to know, what do they want? Do they need mentoring, coaching, training? Just to identify what they're really after. That's the first thing. If they really think they know now they need mentoring, a program would be the best approach if unsure, if they don't have a direct contact person. Otherwise, I think reaching out a quick email would be sufficient, but it has to be really specific, because the working people take time out of their work time of the day to make time for mentoring the young ones. So it needs to be really efficient then.
Yes, wonderful. And I want to ask one last question to you, Alex, and maybe also to you, Sandra. So when you, as academics, we are very much focused on academia, but I see that you also collaborated a lot with industry. How did you make this connection? Is it that you approached the company yourself, or did you go to conferences to meet with different industrial partners, for example, to make this connection? How did you get to establish this collaboration?
I'd have to say, because of where we were in Oxford, we often had industry partners come to us. And I guess now that I'm in Australia, I'm really trying to go the other way and trying to make collaborations with industry partners. Yeah, it's actually probably not just attending conferences, but attending conferences where you know that there's going to be industry partners. So in Australia, it's there's a conference for just biotechnology, so Oz Biotech. So if you want to see companies, you go to those companies, or you find in Australia you have people that are called the connectors. So they connect industry with academics. So try and get in front of those people to try and make that connection.
All right. Anything to add to that, Sandra? Maybe from your own position now, do you have any tips for anybody who wants to collaborate with industry?
I think the main thing is we get so many requests. Is like, be clear, concise, precise. Don't send 20 pages. Send very short. Have a clear plan and a path forward. That will be really helpful.
Right.
I would also have to say that I think people in academics are being pushed to make industry collaborations because the universities think that that's where all the money is. If you're going to make a connection, you need to be offering something to the company, and the company needs to be giving you something very direct. So make sure you're very clear about what you want to get out of the relationship and what the company can get out of the relationship. Because if you just say, "Oh, just give me some money," it's not a great way to start.
Not gonna work.
No.
Never gonna work.
No, I can imagine. Okay, with this, I would like to end this webinar, and I want to thank Alex and Sandra both for your excellent and wonderful presentation. It was very insightful, and I think a lot of early career researchers can learn from this, from your presentations and from your paths. So for the different people who joined the meeting, we also recorded this webinar, so it will also be available later on the ISV website. So if you want to look it back, watch it back, yeah, there will be an opportunity for that. So everybody, thank you very much for joining this webinar, and again, thank you very much, Alex and Sandra, for presenting, and we hope to see you all in the December webinar again. And everybody have a good day, and I hope to see you back again. Bye-bye.
Thank you. Bye.